The final presentation of this morning is ILIAS Biologics, Inc. from Daejeon, South Korea. Presenting for ILIAS Biologics, Inc. is their CEO, Taejin Ham. Thank you.
Good morning, everyone. This is the final presentation of the morning session. So thank you very much for staying with me. My name is Taejin Ham. I'm the CEO of ILIAS Biologics. ILIAS Biologics is focusing on the development of exosome-based therapeutics. And exosome, we believe, can become the game-changing new treatment modality. I'm going to explain why. As you know very well, all biological pharmaceutical products show their biological activity by interacting with the specific targets of the cell. And the targets can exist either outside or inside the cells. Interestingly, small molecule drugs can be developed for both extracellular and intracellular targets. However, large molecule drugs cannot be developed for intracellular targets simply because they cannot go inside the cells due to their large molecular size. So, well, this is currently an undruggable area, we can call it. Now imagine what if we can have a tool that enables us to do that. It's going to open up a huge untapped opportunity for new drug development, I believe. And exosome can be the key to this solution because exosomes are natural nanoparticles secreted by living cells, and they are generally non-immunogenic and non-toxic. They also have excellent biostability, and they can cross various biological barriers, such as BBB. And because of these advantages and their ability to transfer informational compounds, such as proteins, lipids, and nucleic acids, there has been a huge interest on exosomes as the next generation drug delivery system. And now, you may think of a lipid nanoparticle at this point because the lipid nanoparticle also has gained huge interest in the recent years. But, well, it has its own downsides because lipid nanoparticles are synthetically made particles. It induces immune response, and sometimes it can be toxic. And also, it has a lower biocompatibility compared to exosomes, as well as lower intracellular protein delivery efficiency. On the contrary, however, exosomes have excellent biocompatibility, stability, and immunogenicity profile. And because of these numerous advantages of exosomes, as you can see here, there has been an exponential growth of exosome-related research in the recent years, demonstrated by both the number of patents and journals on this topic. And well, big pharmas are also noticing this, and they are paying also increasing attention to this area. So, for example, in 2020 alone, there has been multiple deals signed between exosome specialists and big pharmas, including Eli Lilly and Takeda, and total deal size in that year was about $10 billion, and the individual deal size was about $800 billion, which is quite significant. So what do we do with exosomes at ILIAS? We have developed 3 platform technologies which can potentially revolutionize intracellular protein delivery. Our first platform, EXPLOR, enables the loading of large molecules inside the exosome in a completely free form. I will explain a bit more in detail later. Our EXO-Target technology enables the active targeting of exosomes to specific organs or tissues. And our Pure-Exo technology allows the manufacturing of exosomes in a pure and consistent quality at a very large scale. And then, well, what's great is we can combine these 3 platform technologies to create a lot of novel exosome-based therapeutics. So now let me explain a bit more in detail about our first platform technology, EXPLOR. As you can guess from its name, well, this technology leverages optogenetics. We use light-sensitive protein modules called the cryptochrome 2 and CIBN. And these proteins bind to each other under light with very specific wavelength. And they dissociate freely when the light is removed. So we take advantage of this system by making two fusion proteins in the producing cells. So first, the protein of interest is fused with the cryptochrome 2, and they stay in the cytosol. And CIBN is fused with CD9, which is the exosome targeting protein. And during the cell culture, we provide blue light, and these two fusion proteins combine to each other through the natural biogenesis process. And this results in the incorporation of the cargo protein in the inside lumen of the exosome. And after the exosome is harvested, we remove the light, and then the fusion protein modules dissociate, leaving the protein of interest in the free form inside the exosome. So this is quite a creative idea, a revolutionary idea, and we published this in the journal Nature Communications back in 2016. And now we have registered patents for this in most major countries, including USA, China, Japan, and Korea. We call our exosome technology as fourth generation, and this is clearly differentiated from other conventional exosome technologies. For example, first and second generation technologies, they cannot carry large molecules inside the exosome. They either carry no active ingredients or small molecules in it. And third generation technology can include proteins in the lumen of the exosome, however, only with the linker permanently attached to the inside the membrane of the exosome. On the contrary, however, our fourth generation technology allows the loading of the cargo protein in the completely free form inside the exosome. So this is very important difference because this results in the enhanced therapeutic effect of the same protein once the exosome is uptaken by the target cells. And one of the beauties of our platform technology is its broad applicability. We can load macromolecules up to the size of 168 kilodalton, basically meaning that we can apply this technology to pretty much all kinds of therapeutic proteins, including transcription factors, intracellular antibody, and even gene editing machines. Maybe the most fascinating and exciting application of our EXPLOR technology could be this one, targeted protein degradation, also known as PROTAC. We have very exciting data demonstrating that our technology successfully delivers biological PROTAC into the target cells and successfully delivers and also shows intended effect. So this will open up huge opportunity, we believe, to develop a lot of new drugs leveraging protein degradation technology, especially targeting specific targets inside the cells. And we gave a trade name, EXOTAC, as you can see here, to this particular application of our technology, because it has various advantages over conventional chemical-based PROTAC or biological PROTAC, such as unlimited repertoire of target protein and efficient intracellular delivery and minimal off-target effects. Now, our second platform technology, EXO-Target, this enables the active targeting of exosomes to the specific organs and tissues, as I said. If we do not engineer exosomes, they usually go to the kidney, liver, and immune cells due to their natural tropism. But we could engineer our exosomes by displaying tissue-specific sequence on the surface. And in one of our proof of concept studies, we could dramatically increase the exosome delivery to the brain. We displayed the BBB-specific target sequence on the surface of the exosome, and as you can see here, this resulted in approximately 30 times increase in our exosomes BBB penetration. So we are very excited about these results. Last but not least, our Pure-Exo technology is our advanced process to manufacture GMP-grade exosomes, and we have applied this technology to manufacture more than 10 batches already. We can manufacture at the 100-liter scale currently, and we are in the middle of the scaling up process to up to 1,000-liter scale. And through this process, we could manufacture exosomes with a very high purity, higher than 99.9%, and also excellent batch-to-batch consistency in terms of the consistent quality control criteria. Now, pipeline. As I explained, our platform technologies are so versatile, it can be applied to, theoretically speaking, infinite number of macromolecules and target diseases. So we are very careful in selecting disease targets when we build our own pipeline. So our first lead asset, ILB-202, has the NF-kappa B inhibitor as its payload, and therefore it has anti-inflammatory effect. It has shown excellent in vivo efficacy in various inflammatory disease models, such as acute kidney injury, pancreatitis, preterm birth, and sepsis included. And we recently secured the regulatory approval to start our human clinical trial of ILB-202 in Australia. And this approval is quite meaningful, I think, because it is the first of its kind as an engineered exosome with the systemic administration. And also, we are building pipeline on oncology and CNS, and we are doing this with partners whenever it makes sense. For example, recently we signed a research agreement with the two top-tier Korean biopharmas, JW Pharmaceutical and HK Innoan. We are running our business on 3 business models. First, we are seeking partners very actively who want to leverage our platform technology to develop exosome-based therapeutics. And we will continue to progress our own pipeline and be open to licensing out the asset when there is the right partner. And lastly, we launched a contract analytical service for exosome-based products recently, and we call it Evaluation Square. And our clients can benefit from the state-of-the-art analytical service that we have developed. Now, let me close my presentation by introducing our team and also our company's financial status. Dr. Choi-Hee Choi, who is in this room right now, is the founder of the company, and he's a neurologist by training. He served as a professor at KAIST, which is kind of like a Korean version of MIT, if you don't know. He invented the foundational technologies of our company, and he is still very actively leading all scientific efforts, along with the 3 other very experienced scientists who came from great companies like Amgen or SK Biopharm. On the left-hand side, on the business side, right-hand side, I am leading the company as a co-CEO. I'm a pharmacist by training, and I have MBA from the Wharton School. I worked for a big pharma like Eli Lilly and Celgene in various leadership roles prior to joining ILIAS 3 years ago. And I am working with a team of great professionals in the area of business development, finance, and intellectual property management. So our company has raised $55 million so far over the last 7 years, and we are in the middle of a Series C funding. With the planned clinical trial starting very soon, actually this quarter, we hope to raise an additional $10 million to successfully complete the first clinical trial, as well as the subsequent trials that we are planning and to be finished by the end of 2024. The ILIAS Biologics is a small biotech, but the impact of the technology that we are developing is not small. And more importantly, the passion that we have for bringing this new treatment modality into the world is even greater, I believe. Thank you.